Well, now let us talk about the remaining single agent that is used for treatment of leishmania. And this remaining agent is in form of uh, oligosaccharide antibiotic, which we call it as paramomycin. It is also sometimes referred to as aminoglycoside. So the anti-leishmanial activity of this paramomycin was demonstrated back in 1960s, and this particular paramomycin component was isolated from Streptomyces christomucitus. Streptomyces christomucitus. God, that is why I left microbiology. Anyhow, that was isolated from that scary spelling species. Originally, that paramomycin was designed to use to treat intestinal infections such as amoeboisis and cryptosporidosis. So, this paramomycin is provided with a trade name of paramomycin sulfate, and the drug delivery route is by oral way. Okay, well, now let us talk about the mechanism of action of this paramomycin. So, still we have not clearly understood how this works in Leishmania, but still two mechanisms are proposed that tries to explain how this paramomycin can function as a leishmanicidal agent. So, mechanism one involves inhibition of protein synthesis, while second mechanism involve induction of apoptosis. Okay, well, let us discuss both of them one after another. So, in mechanism 1, that is inhibition of protein synthesis, this aminoglycoside antibiotic is said to bind and affect the normal ribosomal function. Okay, so now as you know, the bacterial proteins or the proteins are synthesized by ribosomal RNA complexes, which are composed of two subunits, the large 50S subunit and small 30S subunit, and collectively these two forms 70S subunit, as you can see in this figure. So this 70S functional ribosomal unit has three functional site. We call it as E site, P site, and A site. So this A site is where new incoming tRNA binds to mRNA. It is this P site where the ribosomal uh, this P site is a ribosomal site, and it is most frequently occupied by the peptidyl tRNA, that is the tRNA carrying the growing peptide chain. It is where the peptide bonds are formed and eventually the polypeptide is elongated. Next, we have E site or empty site that holds the tRNA that has just give up the amino acid. So, what happens now is that this paramomycin might bind to 30S subunit that consists of 16S rRNA and the S21 ribosomal protein. Well, now one might argue over here that this 16S rRNA stuff is specifically present in bacteria, and uh, this parasite being eukaryote, how these things might happen. So, here lies the very rub in this thing. So even though the exact 16S rRNA is not present in eukaryotic system, but its analogous molecules are usually present that can carry out the similar function. So no cell can function without the activity that is similar to 16S rRNA. So this binding of our antibiotic might cause defective polypeptide chain to be produced, and it involves something called as codon misreading, due to which some inappropriate amino acids are attached to the growing polypeptide chain, and due to such continuous production of defective protein, eventually that would lead to pathogen death, because once the primary sequence of protein has changed, it won't fold properly. If it won't fold properly, it won't function properly. It would create defective protein, and due to defective proteins, the organism may not survive properly. So that is one of the mechanism. So this is how the summary of all of the agent that we have discussed. It can be summarized in this single slide. Pentamidine, it can be uptaken by the transporter. It can inhibit the polyamine uptake and arginine transporter that might lead to anti-leishmanial activity that we have already studied. The pentamidine might be taken up by transporter. It might get accumulated inside it and associate itself with the kinetoplastid DNA. The kinetoplastid DNA association with kinetoplastid DNA might affect the binding, might affect the replication system by inhibition of topoisomerase 2 activity. Due to inhibition of topoisomerase 2 activity, apoptosis like death might have happened due to nuclear condensation and DNA fragmentation. This might lead to the anti-leishmanial activity. On the other hand, the same pentamidine can inhibit the components of your respiratory complex that might result in disturbances in mitochondrial membrane potential. Due to differences in or disruption of mitochondrial membrane potential, it might lead to apoptosis like cell death, which might include nuclear condensation, DNA fragmentation, and eventually the parasite would die. In case of miltefosine, it can target the sterol content, or it can decrease the mitochondrial membrane potential and lead to anti-leishmanial activity. In case of parabomycin, it is suggested that it might bind to the A site of rRNA or to the ribosomal subunit that would result into misreading of mRNA and inhibit the recycle of ribosome, and this might result in anti-leishmanial activity. While the parabomycin can also affect the mitochondrial membrane potential and eventually lead to anti-leishmanial activity. So this is how all these things can be bound together and explain how these therapeutic modalities like pentamidine, miltefosine, and parabomycin might work. So this is in the nutshell. Now I would advise you to read these two papers to get more insights into what are the uh, therapeutic options which are available for management of this disease and their molecular details involved therein. Okay, so now, well, let us move our discussion from what are the current therapeutic arsenal that we have to fight against this disease to how we can use bioinformatics for designing novel therapeutic opportunities, right? So I would demonstrate you how in silico techniques like subtractive genomic approach can be used in target identification, and we would have leishmania as one of our model organism. So, in order to understand the subtractive genomic approach, you must first understand what is metabolome. So, metabolome is a term that describes, or you can say, that constitute the entire metabolic content of an organism. So, in short, you can say just like proteome, which states the entire protein content of a cell, metabolome is in similar way the entire metabolic content of your cell. And as you can see in this picture, the metabolome is very complex. You can see that there are a lot of metabolic pathways, and these metabolic pathways are associated with each other. Everything is interconnected, and everything affects everything else. So this is how we define a metabolome. Well, now what we will do in subtractive genomic approach is that we would compare the study, or you can say we would compare the metabolomes of two organisms, that is Leishmania and your human host. So the basic fact, or you can say the basic principle behind such study, is that we know that organisms evolve independently, and hence they do differ biochemically. This would mean that not all organisms would contain exactly same biochemical pathways and same biochemical reaction. Some of the reaction will be present in one organism, while some of the reaction might be absent in another organism. Okay, some organism, we'll say for example in case of our parasite, it might have evolved some entire different set of biochemical reaction and entire different set of metabolic pathways, uh, that the host organism might not use the same pathway to create the same metabolite. So all in all, what I want to convey is that in due course of evolution, when these pathogen and your host evolved independently, they have figured out their own way of doing the same thing. Okay, and here lies the opportunity. So for comparing the two metabolome, or what you can say the objective of comparing such two metabolome would be to identify the reactions that are unique to pathogen. Okay, so when we say that we are going to compare two metabolomes, the objective is to classify the reactions which are present in both of the two species into three categories. First category would be the one which would have all of the proteins that are present, or all of the reactions or metabolic pathways that are present in both the organism, that is your pathogen as well as that is your human. Another category would be the proteins, or would be the enzymes, or would be the metabolic pathways or biochemical reaction that are uniquely present in human. And the third and final interesting category would be a set of biochemical reaction, a set of metabolic pathways that are uniquely present in your uh, pathogen. So in this way, enzymes of such reactions which are uniquely present in pathogen forms a very attractive drug target. Okay, so besides uniqueness, if your identified drug target, or any protein or enzyme, if it is if it catalyze something called as a choke point reaction, it would be very beneficial to act as a drug target. You should also make sure that whatever protein or enzyme that you select as a drug target should be vital, or you can say it should have a very essential function in pathogen physiology. And finally, whatever drug target you select, it should not have close homologue in host genome. That is, the very first criteria was uniqueness. That means the same protein should not be present in both the organism. And the fourth criteria is about homologue. Homologues means the proteins which are not the same but which are related to each other. Well, those are included in homologues. So well, let us discuss all of this aspect one after another. First, let me tell you what are choke point reaction. So to define a choke point reaction are such reaction in your metabolic network that either uniquely consumes a specific substrate or produces a unique product in the metabolic network. Okay, so the enzymes that are involved in choke point reaction are termed as choke point enzymes. Okay, so to give you the visual taste of this thing, I'm showing you with a figure wherein I have highlighted the choke point reaction in red. Okay, so in this figure, you can see the reaction that is highlighted in red. This first reaction uniquely consumes a specific substrate. Well, this would eventually mean that this reaction is responsible for consuming a substrate that is produced by multiple metabolic pathways, or by multiple reactions, or multiple biochemical reaction that produce a single metabolite. Okay, and this single metabolite is further consumed by this enzyme, and in such a way it is called as a choke point reaction. On the other hand, you can see over here, in this case, the highlighted red colored reaction, it is responsible for producing a metabolite that is very unique. That means it will produce a metabolite that will form a source of multiple metabolic pathways, or that would form source of multiple biochemical reaction, and eventually they would form multiple metabolic pathways. So now, if we inhibit such enzymes that uniquely consumes a specific substrate or produces a unique product, what would happen in this case, as you can see, since it uniquely consumes a specific substrate, इससे होगा क्या? That would result in accumulation of a lot of metabolite that are consumed by a specific reaction. Okay, and accumulation might result in toxicity related death of the parasite. On the other hand, if you target the enzyme that produces a unique product, in such case what would happen is that since this enzyme is inhibited, it would not be in the position to generate a diverse set of intermediates that are supposed to be subsequently produced after this metabolic reaction, and therefore the pathogen would die off due to scarcity of lot of metabolites that were supposed to be produced from the product of this unique reaction. Okay, therefore, inactivation of such enzyme can choke a specific metabolic pathways in which they are involved, and that would pose a threat for fitness or survival of that pathogen. Therefore, a choke point enzyme serve as a very good potential drug target candidate. Okay, so now let us talk about the concept of vitality and non-homology. So, in case of vitality, it is essential that the enzyme should perform a very key role in the pathogen physiology. Okay, so that if you inactivate that enzyme, the parasite should surely die. Why? Because unless an enzyme holds a key position in pathogen physiology, its inactivation will have no effect. Hence, the drug target enzyme should be vital in pathogen physiology. To just give you an example, uh, one might think, I would give you or cite you an example of targeting your nucleotide biosynthesis pathway. Okay, so there are two pathways by which your nucleotides are produced in host cell or any cell as such. We call it as de novo pathway, and we call it as salvage pathway. So in de novo pathway, it involves a set of biochemical reaction which are responsible for construction of nucleotides which are responsible for construction of nucleotides from scratch. That is why we call it as de novo pathway. And in salvage pathway, whatever degraded component of nucleotides are there, they can be joined together to form the nucleotides as such. Okay, so now one might think that if I inhibit the enzymes from de novo pathway, it would act as a good therapeutic opportunity, because it would eventually hamper production of nucleotides नहीं रहेंगे, न्यूक्लिक एसिड्स नहीं रहेंगे, न्यूक्लिक एसिड्स नहीं रहेंगे, डीएनए आरएनए नहीं रहेंगे, तो पैथोजन would die. But in such case, even though if the de novo pathways are inhibited, the pathogen has compensatory pathways in form of salvage pathway, which eventually it can use and create the same nucleotides. Therefore, you cannot assertively say that this de novo pathway constitute the vitality or the role of vitality in such scenario. Therefore, you should target a protein that should be very vital, that should play a very key role in pathogen physiology. It should be a sure short way, so that if you inhibit it, there is no way for that protein to survive. There are no compensatory pathways that would create the same metabolites by some another different route. So this is the concept of vitality. Okay, now let us talk about non-homology. So the human genome should not contain any close homologue of the identified target protein. Okay, so if you make assurance that there are no any close homologue of the identified target protein in host, so that it should not so that your host should not be curtailed by drug that is identified to inhibit the pathogen enzyme. तो ऐसा नहीं होना चाहिए कि whatever protein or enzyme X that you have identified in your pathogen and you plan to use it as a drug target, it has got some closely related protein X prime which is present in your human host as well. So since this X and X prime are now homologues of each other, they would share sequence similarity as well as structural similarity. And when you design drugs to target this protein X, since your homologous protein X prime, which is present in your human host, would also be affected by the same drug molecule. So this is what we call it as off target toxicities. So usually uh, the proteins which are close homologues to the identified drug target are usually the off target proteins that can be hampered by the same drugs, and it won't be a very good scenario in your drug discovery campaign. Okay, so now it is very essential that whatever drug protein that you select, you should make sure that its close homologue should not also be present in your human host. So these are, you can call it as the four criteria. Okay, so just to summarize it, you can use the subtractive genomic approach to compare the entire metabolome and identify the unique enzyme. Then you can make sure that whatever unique enzyme that you have identified, it would be better if they follow a choke point reaction or they catalyze the choke point reaction. They can be choke point enzyme. Then you can make sure that whatever choke point enzyme that were unique, they are supposed to be vital. Next, whatever vital choke point and unique reactions that you have identified, they should not have any close homologues in human genome. So in this way, uh, what things would happen is that when you start out comparing the two metabolomes of human as well as pathogen, you might end up comparing a few thousand biochemical reactions or a few hundred metabolic pathways, and after comparing you might end up with identifying say some 100 unique reactions. Okay, from these 100 unique reaction, you would shortlist say say for example of these 100 unique reaction, maybe there are only 70 enzymes that catalyze this choke point reaction. Then you would again shortlist out of the 70 few set of enzyme, it can be say 50, that are very vital for survival of that pathogen. And eventually of these 50 you might end up with say 5 or 10 enzymes that do not have any uh, homologous enzyme to your present in your host genome. So in this way, at each and every step you go on subtracting, or you can say you go on shortlisting the number of enzymes or the number of target proteins that would form your putative drug targets, right? So in this way, since at each and every step, the number of the target protein would go on eventually decreasing, and hence we call such approach as subtractive genomic approach for drug target identification. So, since the number of these protein do decrease with each and every step, some people also refer it as a reductionist approach for finding out the drug target. So, whatever the name is, the concept would be one and the same. You would begin with a large number of enzymes, and for each and every test that you conduct, you would go on subtracting, you go on reducing, or you would go on discarding the number of enzymes or number of drug targets that you are considering, and end up with very small number of possible final drug target enzymes. Well, this approach is collectively called as subtractive genomic approach. Therefore, now let us talk about what softwares can you used in such drug target, or you can say subtractive genomic approach for identifying drug target. Well, there are online tools like Rehnuma that you can use for comparing the entire metabolomes of two organisms. Using this online server, you can identify which are the set of reactions which are unique to your pathogen. Then whatever unique reaction that you might have identified, you can copy those list of EC numbers of those unique reaction and perform choke point analysis using an online software, using an online server called as MetExplore. This MetExplore would then further tell you of the unique reaction that you have provided, or the enzymes that you have provided that catalyze those unique reaction. Of those enzymes, how many enzymes follow the choke point reactions. Well, this would be included in your choke point analysis, and you can do this using software called as MetExplore. It is an online tool. Then once you end up with a few choke point enzyme, then you might start thinking on like of the choke point reaction, how many are essential or how many are vital. For that, you would need to perform the essentiality test. So people usually perform this essentiality test by doing the blast based search in database of essential gene. So let me now first tell you what is this database of essential genes. So what the creators of this database has done is that they have tried to compile a set of genes that are known to be essential for maintaining the life form. Okay, so what are the minimal set of genes which are essentially present in almost all of the life forms that we know on Earth, and the compilation of that genes, they have called it as database of essential genes. So this database would contain a set of genes which, if they are absent in any organism, that would result in fatality of that organism. Okay, so this database of essential gene contains a set of gene that we know are important to sustain life on Earth. Okay, so now what you would do is that whatever enzymes that you have obtained from choke point analysis, you would try to do a protein based blast, and if you find out that the ortholog or the similar protein already exists in your database of essential gene, that means that protein is also essential in your pathogen, and hence you decide the essentiality or the term of vitality in your pathogen. This is how you perform the essentiality test. Okay, now the final test that you would be interested in performing would be to decide non-homology. It, as I have already told you, it should not happen that any close homolog is to be present in your host genome for whatever target protein you have decided. So what you can do is that you can perform a series of Cyblast result, or you can perform a series of Cyblast searches. And as you know, as you are already aware, that Cyblast is used for detecting the remote homologs if possible. So what you would do is that you would perform a series of Cyblast taking the sequence of your target protein, and you would perform the blast searches in human genome. And if it happens that after performing a few iteration of Cyblast, if you still don't end up getting any hits, that would mean that your host genome do not contain any close or distant homolog for the given target protein, and eventually that would be your best set of target proteins that you might have obtained from such subtractive genomic approach. So we have performed such study, and we have I would just let you know the statistics that we have recently published in our paper. So we have exactly implemented this approach, and here is how we have obtained the result. So when we compared the entire metabolomes of Leishmania with that of human, we end up comparing 1627 reaction, from which we identified that there were only 97 that were found to be unique in the pathogen metabolome. Some 1036 reaction were exclusive to a human, and remaining 492 were common in both. Therefore, we further focused on those 97 reactions that were found to be unique in pathogen metabolome. So next what we did was of this 97 unique reaction, after doing choke point analysis using MetExplore, we ended up with 73 choke point reactions. And further essentiality test that I have already told you perform on that DEG server, we shortlisted again 14 enzymes that are vital for pathogen survival. And finally, after doing a series of Cyblast searches in host genome, we ended up with 6 enzymes. And now we say that these 6 enzymes are thus represent the appropriate targets for Leishmania. So now the bottom line is that subtractive genomic approach provides a theoretically sound drug targets. Okay, so we can assure that if we target these proteins, theoretically it is possible to design a better uh, intervention therapy as such. Okay, so you can learn more about such subtractive genomic based target identification as well as metabolic simulation in the paper that we have published in Parasitology International in last year. So it would be your assignment to read this paper, understand the concept of subtractive genomic, and appreciate how bioinformatics tools can be used for identifying new drug target. We have considered Leishmania as one of the model organism. You can implement, you can start thinking of implementing such approaches for your model disease that you might be working in your project. So so this can be a good start for you to start thinking in an innovative way, wherein you can end up finding out new drug targets that would eventually help you getting a good publication right from your project work. Okay, so this is it. We have already discussed this slide. Thank you very much.